this. Okay. Hello everyone. I'm very honored today to host Professor Luonan Chen uh, of the Shanghai Jiaotong University. And Professor Chen holds a chair professor in between the School of Mathematics and School of the AI, with the background includes the BS from the Hwajung University of Science and Technology and PhD from the Tohoku University in Japan. He has built a distinguished international career. His research lies at the intersection of applied math, information science, and AI and life sciences. He has pioneered the new field of dynamical data science and AI-enabled science, focusing on dynamics, network, causality, tipping point, prediction, and AI method for complex biological and medical system. So he has published more than 400 scientific papers in top-tier journals such as Nature, PNAS, Science Advance, and etc. So please join me in welcoming Professor Chen, who will present AI for dynamical systems biology.
Mm. Okay, thank you, Jerry, for the nice introduction. It's also my great honor to talk at this seminar. Today, I mainly talk AI and causal inference. The title is AI for Dynamic System Biology. I gave a new name. We call it traditionally I use the computational system biology, but I think it's not a good name for me. So I changed it to the dynamic systems biology. Okay.
Okay.
Then there are two parts. The first part is the AI. So well known to us, last year, yeah, Nobel Prize awarded to the two distinguished scientists for related to AI field. I think AI, the most powerful um, the the abilities they can represent the high dimensional states. So um, then for this part, I want to represent our static structure, our body. So so I started from here. What's the static structure for our body? Our body we have 23 trillion cells. So um, if we can understand the state of each single cell, and then we can further consider multiple cell as a network. So we can further understand the tissues and then the organs. Eventually, we can quantify static state of our body. Okay, so that is the scheme. We know we have 20,000 genes each cell and also more proteins. And in the textbook, we know we have 200 cell states, I mean cell types. But now, based on the recent, progress, we know there are over 300 cell types now. But in the next year, I think they probably came to the 400 because we are single cell technology, uh, progress so repeatedly. And now we have probably over 500 million cells available, I mean, public available at the transcriptonic level. So with such data, there are so many, related works how we can pre-train this data to get we call the foundation model for quantify static state of cells. And so the first one I think is Genoformer is 2022. They used I think 30 to 40 million cells to train. They got the they get the very nice embedding, so it can use it for downstream analysis. So our CAS, our first work in the Chinese Academy Science is the Genome Canvas, which was available, I think it's 2023, but the paper published last year. So here they used 126 million cells for cross-species, um, you know, um, pre-train the model, the foundation model. And then further, they have AC foundation model. Um, this year, Evo 2, they can even directly use the DNA sequence to represent the cells. And also this year, Alpha Genome, and also DNA sequence, you know, um, model, and it's not so well defined so far. But I think it will be quickly pick up. But now at the RNA-seq level, I mean transcriptomic level, I think all this the model foundation model, they can be used for various tasks. So next one I will show the work of our group. We published the first one is 2022, but
Mm,
is for the multi-cell state. To quantify multi-cell state, we call the STMVC.
mhm,
We use the deep learning, it's a contrast learning with the diffusion process, use the we call the imaging data, and also say this is locational data, further RNA-seq data at each spot with the pre-knowledge of the medical doctor. So we integrate all this four different data then and to represent the steady state um, of the multi-cells. Then last year we further consigned the cell-cell communication. We built another one we call the ST-Caper. So we aim to quantify 2D, two-dimensional tissues for the static states. Then this year we also build another one. We want to make the low resolution data become high resolution at the single cell level. Most of the data so far is at the spot level. With the multiple cells in one spot. Also, the data quality is not so good. So we built this model. And then this year we also build another one. We want to consider 3D tissues and how we can integrate multiple slides to consider organs static state. So we call it SC clinics. So I pass all the details. Is we just use the deep learning technology. So we build also there are several ongoing work, and we build many related model. We want to, as mentioned, we just want to represent the static state of the cells, tissues. So with such a preparation, we can further do the downstream analysis. The one is we do the causal inference. The one, you know, task is we want to do the observational causality, just based on the data. We want to infer, say, how the data affect the phenotype disease. So that is another one is this is
mm,
observational causality. Then we build another one we call the interventional causality. And this one we just use the say observe the data and
mhm,
can build intervened
mhm,
distribution. So I think that is quite I think is a very nice theory. And traditionally so far, this area is dominated by the structure causal model. It's Rubin and Judy Pearl. it works, excellent work. So, but they require pre-knowledge on the network structure. So, but this year we published another one. We called they called backdoor criteria. We called knock-off backdoor criteria. So in this work, we don't require such a pre-knowledge. So, but you can see um, intervened distribution, is quite similar as the backdoor criteria. But here we have a s- twice integral. This is only once. Okay, I will show later about this work. Okay, then we use this is a new interventional causal criteria. Then we build gene regulatory network and also build the relation, causal relation from the genotype to the phenotype. Okay, then we apply to the micro metagenome, gut metagenome, how gut metagenome affect fat liver disease, I mean disease phenotypes. Then we also apply to the with another method for the androgen how affect the diseases. Um, and then I will also like to show how we can quantify our state body use the other type of data. The one is we call the CGM former. I think we build the first such a pre-trained model. We use the say 58,000 subject. CGM is they are cheap, just touch to your body. They can measure glucose level, every 1 minute or every 5 minutes. They can measure around 2 weeks. So each person we have a long time sequence. So we use this time sequence to make the token, then quantify each individual states. Okay, so then we build I passed all the detail for our pre-trained CGM former, so they can for the subtyping and warning and prediction, and also in particular they can do the um, virtual intervention. Yes, it means as far you let me know how much you eat, I mean your dish, you like the vegetable, how much rice, how much, so we can make the prediction of the future glucose level and within 24 hours for each individual. I mean, it's an individual basis. Okay. Then this year we built another databases we called say organoid. The reason we built this isn't my colleague conducted this work. He's an excellent experiment. Why we work on the organoid? The reason is, FDA recently, I think last year, make the new policy for the drug development. We don't need they don't require animal test from now on. So which means we don't need to do the animal the experiment, but then you cannot do the experiment on human. So organoid become very popular. So we build the databases. Then based on this basis, my students build a first one we call the virtual organoid. We call the OD former. For this work, we collect all the available, it's for the pancreatic cancer. So we collect all available pancreas cells, a few millions. Then we also collect the drug perturbed the cell line data, like the linked and also CMAP, and all
Mhm.
available. So we make the pre-trained the model for the drug perturbation. And for drug model, we use the SMART structure. So we pre-train the SMART structure for, I think it's 100 million different SMART structure. Now with this one, this is the pre-trained model. They can simulate the, cell perturbation by the drug, but not human. So then we use our organoid data,
Mhm.
and we for this data, we we built an organoid for 191 patients, I mean pancreatic cancer patients, used 98 different drugs for the perturbation. So the total is we sequenced the 100 14,000 PTC, you know, the gene expressions.
Mm-hmm.
Then with this one, we can align pre-trained model to the human, I mean to
Mm-hmm.
the organoid. So in this way, we can make we can make the downstream analysis. The one is I think the most important one. We can make the personalized medicine recommendation for each individual pancreatic cancer
Mm.
patient. Eh, So, which means that as far these patients have no drug to treat when in the later stage, so we can use the over 100 different small molecules uh, to simulate their IC50, how drug effect for this individual person. We need a biopsy to take the tissue, get the RNA sequence, then we can transform into his virtual organoid for every, small molecular testing. So, in this way, then we test to the three
Oh,
cohort.
so Ronan,
The
so
first,
previously, so here you
mm,
use the two data set, organoid data set and
mm,
tissue data set, right?
organoid.
Only
For
organoid?
organoid, yeah, tissue is organoid is built from individual tissues.
Okay, but here you said the pancreatic cancer bulk RNA-seq, pancreatic
Mm,
cancer SC
mm.
RNA-seq. This is not the organoid data set, right?
Uh, this is not this part is not organoid. This is
Oh,
all available. This is the public available data for pre-trained model.
oh, okay,
Then this is we use the pre-trained cell line data to align to the human data.
okay.
So this is a drug perturbated on the organoid. As mentioned, we used 98 different drugs
Mm-hmm,
to perturbate this
mm-hmm. Okay,
organoid. So we
so
get the
okay, so for the
mm,
about 200 organized, you
mm,
applied around 100 drug, and then you use that data set.
right. Right. So,
Okay.
we use the drug. We have a drug smell structures, and so also have a individual organoid. I
Mm,
mean sequence, RNA sequence. So they are perturbate and after, before perturbate and after perturbate.
mm-hmm,
So then we can collect. Then we can align the pre-trained cell line model to the organoid model. So we call the virtual organoid.
mm-hmm. Oh,
Okay.
okay, so but here you have still pancreatic cancer cell tissue in the right side, 272 patients. So do you use this
Mm.
bulk RNA-seq data from cancer tissue or not?
Uh, this is also used for training. You can use the book. We also have actually we also have books from public. We use the TCGA the
Yeah,
data
yeah.
book. They can use you can use they all use in our the model for the training.
Mm-hmm. Okay.
Okay. So eventually we only require bulk data
Mm-hmm.
for the testing.
Okay.
It's a we yeah. Actually, organoid is a kind of the bulk. It's not a single cell
Mm-hmm.
organoid.
Mm-hmm.
It's kind of it's a and yeah, actually a different cell types in the organoid. So you can consider the organoid is kind
Mm-hmm.
of the bulk. Mm.
Okay, okay.
Yeah, but anyway, you can check. We also put this online. You can check the details. So anyway, so this way the up level is each line is one person. Is one
Mm-hmm.
person. And when we with their bulk RNA-seq data, and then we make a prediction and predict the top 10 drugs, mostly if effective for this individual person.
Mm.
So if they are real, they are drug tricked, use the drug same as our recommended, you can see the survival rate
Mm.
become much better. Otherwise, you can survival rate is significantly lower than
Mm.
them. Yeah. So now we apply the IID, scientists testing for the hospital. Right now is in the progress. Okay. Then the second the foundation model is breed baking. For this one is also mainly conducted by my postdoc. So we use this is for cross-species study, and we use the mass, two type of promise, than one human. So it's a four different species. And then we use the spatially resolved transchromatomic data with the different platforms like the Murfish, Xenium, Stereosec. So we all collect, all available. So total is, I think, is 140 million cells for training. And so this is, as mentioned, is a multi-slice for the, as a brain, for the simulated brain. So then we train, this is a quite a large scale study. So we actually used over 300 GPU for training. I think it's over 1.5 months
Mm.
for the, it's very computation intense. Anyway, and so with this the model, we can do the cell type annotation, label transfer, to one species to another species, and the network, the inference, and importantly, we can do the cell or niche perturbation, virtual perturbation. You can change the cell type, and then we can see how the brain tissues change, with the your
Mm.
perturbation. So we also do the testing, virtual testing,
Mhm.
pass those details. So this is, I think, is the first such a bring, the model at the single cell level. This one we also published another one. Is conducted by master students. So what's this one? This one is we want to one drop of the blood reconstruct the human 3D face. And so is we just use the only genetic information, a sequence then diffuse to the human 3D faces. So human is a 10,000 dot cloud. So each human face is a 10,000 dot plot, dot cloud. So we use the 7,000 the samples for testing, for training, 2,000 for testing. So total is 9,000. So for this samples, we have both the uh, gene sequence and also 3D faces. And then we use the model is the input data is SNPs and BMI because our face is depending on the BMI and age. So with three information as input, we can predict um, this human 3D facial image. So so this one, the interesting is so far, we consider our face is mainly influenced by the genetics and epigenetics. But actually, it's true. But but we found out that the interesting is uh, our faces changed from a baby, small
Mhm.
to the adult,
Mhm.
become big, then
Right.
aging become small. So
Mhm.
our face change with time. But you are found few point in your face,
Mm.
their topological structure is
Mhm.
decided by genetics. So that's very interesting. So, but with our diffusion model, you actually can capture such a topological structure. Then eventually they can rebuild your structure, human faces. So with this model, we can, as far you give me, uh, say, genetic information like the SNPs, then we can, and for instance, like the baby, um, as far you have a gene sequence, then we can predict their face at 18 years look like this one, 45 look like this one, 80 years look like this one. We can simulate the whole life, the image. Um, yeah, then I think the last part is that I also, because I mentioned we want to use AI to represent the static state of human cells, tissues, organs. So traditionally, besides the AI, besides the deep learning, we also have many other approaches like PCA. It's a linear. We can reduce dimension to 2 or 3 dimensions. We can represent your high dimensional data, or TSNE, UMAP, many others. But all these methods, say, is very nice, but they have problem. They cannot preserve original data topological structure. So, which means as if if you transform the original high dimensional data to the embedding space, they may lost their original topologies. So, in this work, we build another one model, method we call the DTNE, and we can preserve or original topology even in your embedding space. So I think it's better to promote this work. So we also comparing to the PCA UMAP, and to show it's better.
Hmm.
Okay, and so I think and today I mainly want to show say uh, the causal relation. So I want to move to the
Hmm.
our main topic. And so I passed all the tipping point and also STI equation. I think today I want to show a little bit how we can um, um, we have another slice. Slice is um, this one is why we have another one. Um, is quantify sorry, is can you see? No. Oh, we have not yet stopped
Yeah,
sharing,
please
right?
share the, yeah.
Uh, but I, why is not coming? Uh, we have um, okay. First is one. This one I just show you a little bit, but I want to go a little bit detail. Uh, Sorry, today I have another meeting. I have to do quickly. Sorry.
Hmm.
We have- so I
But
okay. Uh, so
we are seeing the like a
mm.
tour screen, not
Oh,
the
you can see
uh
the
but
screen
but screen is
now.
too small.
It's small.
Yeah, because
Oh,
yeah, I think it's in
it's
tour mode.
small. Um, it's small. Why? It's small. I think. I think maybe it's. We can share in this way. Um, we can do share. How about this one?
Still we see the your face
It's a
and
full screen. It's a
uh
full screen.
no, it's a you and
Oh,
light. Yeah, both are up here.
oh, why? It's
Mm.
I think
Yeah, now it's
for
okay.
me it's a full
Previous one
screen.
was okay. It was a good. Just
Right now it's okay.
yeah, yeah, yeah.
Oh, okay, good, good.
Uh,
So I
but it
start. Can
disappeared.
you see this? Huh?
It disappeared. Can you try one more?
Disappear. Mm, how about this one? Um, um, maybe I it's um, maybe I should. I see. I think I first I have to
Yeah.
stop, stop one, stop this one. I think I have to stop this one. Uh, then um, I think I also stop this one. Mm, or it's a bit mm, complicated. I have to stop this one. Then close this one. I only leave one. I think this should be okay right now. Leave one. How about now? Oh,
No.
why is it two? Why
Yes.
is it two
Yeah.
screen? Uh,
So but can you do the like a slideshow first?
oh,
Let's
how
tr
about this one?
No, it doesn't work.
Is that still have two?
Yeah.
Um,
Uh,
mm,
Someone say that can you check the sharing option thing?
sharing option. Uh, sharing. Um,
Or
oh, a whole screen. Oh, I see. I use the whole screen. How about this one? Oh, it's still
oh,
not.
okay. It was okay. Just last minute it was
Mm,
um or
I
can
think.
you what is the best way? If
Mm,
you uh,
um,
uh, what if then we go with a small slide?
Ghost Master. Okay, I closed all of the file. Then they came to um, I, I see how this if this work. Um, um, sharing whole screen, sharing whole screen. Um, oh, I see. Now I think it's okay.
Mm-hmm.
I think it's okay.
Mm-hmm.
Mm. How about now?
Yeah, good.
I see. I think you're right. The option,
Mm-hmm.
there is the option. Mm,
Okay.
okay. So sorry.
Mm-hmm.
I want to show the causal inference. The first one is um, we call the interventional causal inference by knockoff operation. So this is mainly conducted by my master students. Mm. So what's the knockoff operation? Knockoff operation is um, we have a node GK and all remaining nodes. We have a network of say maybe hundreds of nodes. Then we want to focus say the causal node and um, then the effect node, right? Xj, Xk, right? Then for this one, we want to say mm, we want to build we call it knockoff node. Xj is the original node. We want to build another one we call the Xj head node
Mm-hmm.
to replace Xj. So then build this node. We call the knockoff node. Then we need a condition. This condition is um, say,
Oh, oh,
uh,
Lona, I don't understand. So could you explain one more
mm,
time what is the XJ and XJ knockoff?
maybe I use this one.
Yeah.
Say, we have
Uh-huh.
X and Y. X is, you know, um, the causal node.
Mm-hmm.
Uh, Y is the effect node,
Mm-hmm.
right? And we assume X. We want to infer the causal relation from X to J to X to Y.
Mm-hmm.
Z is all remaining nodes, remaining
Mm-hmm.
variables. So we know there is a
Mm-hmm.
theory we call the
Mm-hmm.
backdoor
Mm-hmm.
theory, backdoor criteria,
Mm-hmm.
as mentioned, which mainly conducted by the Judy Pearl. So, as far you network are available, you have a pre-knowledge of a network. You just cut off all the backdoor
Mm-hmm.
rules. For the X, so you can build intervened distribution. Why? After intervention, you do the X, you can have the distribution of Y. Uh, just use the say observed data. So which means that you can use the observed data get intervened distribution. So if this intervened distribution is different from your um, original distribution, observed distribution, which means it's a causal relation. If it's the same, it's a non-causal, right? Because you change the x, there is no change of y, so we call it non-causal. If you change the x, y changed accordingly, we call it causal relation. So that is a backdoor criteria. It's very popular right now, used in the deep learning. But there is, as mentioned, uh, uh, you can infer, this is a conditional, uh, uh, say probability, but they can be right in this way. So we can just use the observed data to get the intervened distribution. So it's very nice, but the problem, as I mentioned, you have to know the pre-knowledge on the network. So then in this work, we build another framework we call the knockoff backdoor criteria. So in this work, as mentioned, you have a node X, but we want to add another one, X head. We just add another one. We are going to build another one. So with this one, then we can get another network in this way is equivalent. So in this way, we don't need to require any network pre-knowledge. The reason is that we build this one, we know the backdoor X hat. So in this way, X is built based on the X hat based on the X. So which means that we change the X to X hat. Then
Mm-hmm.
we can see intervened distribution. Then you can actually mathematically we can prove they can build in this way.
Mm-hmm.
As far as build in this way, we can just use the observed data to make this intervened distribution.
Mm-hmm.
So this is the scheme, and then we can theoretically we can prove is intervened. We call the intervened distribution. Beautiful this way. So I think this is mainly this is the main theoretical part. Um, then use this way, then we can infer the network structure.
Uh,
Because we don't need a pre-knowledge, we can just based on the data, you
uh,
can uh,
so Lona,
check.
okay, so
Uh,
after you make the X tilde, and then
mm.
how do you use it for the causality detection using the X tilde?
Oh, this is it's originally the X, right? They have a distribution, right? It's observed. As mentioned, we already observe X and Y, the data, right? Then which means we can get the conditional distribution, uh, condition on x, observed x. Then uh, right. Then now we changed x to x tilde. Then we can get another distribution.
Mm-hmm.
This is the intervened distribution because we
Okay.
changed the x to x tilde.
Yeah.
So if this distribution is different,
Mm-hmm.
causal relation is the same. There's no causal relation because we changed x to x tilde. There's no change. So uh, uh, so is so I mean causal criteria become just compare these two conditional distributions
So in your
before and
previous
after the interval.
slide,
Mm,
in your
mm,
previous slide before this theorem, next,
yeah.
yeah, next.
Mm.
So this is your
Yeah, this is the
comparison.
method. This method we used here. We used it's uh, just a KL, uh,
Mm,
criterion. But you can say you can use many different criterion. You
okay.
cannot. It's not necessary use the information to do this one. This is just one case. We just use the information.
Mhm.
It's care
Okay, okay.
divergence. So, but it's not necessary.
Mm,
It's
uh, okay,
in
so
principle, yeah,
okay.
you just
So
compare to distribution. Mm.
so you compare the two distribution
Mm.
given x and x tilde, and if they are different, you claim
Right,
that
right.
there is a causality.
Right, right.
Mm,
Because as mentioned,
okay.
it's kind of a backdoor criteria.
Okay.
So
Mm,
it's and I forgot to mention how we build X tutor. You know, build X tutor is straightforwardly. You build X tutor. You want to maintain X tutor and maintain the say association with all remaining variables as the same as the original association. So
mm,
covariance, we have to keep the covariance um, is the same for any variable. You want to keep the same.
mm,
So, but except y, we don't need. So you build a executor, you ignore y. Then, but you have to keep to maintain the covariance with all remaining variables. So in that way, even you change the x, you don't affect any other remaining variables. So they possibly affect y, but we don't know. So we have to calculate. So that is in simply you use this way, you can for each variable you can build their knockoff variable execute. Yeah. So then another criterion you have to want to make the executor as different as possible with the original X. So you have to make them covariance as small as possible. So you can make a big perturbation. So that is the two criteria. You want to make a
yeah, yeah,
executor as
okay. Yeah, yeah.
different as the original X
Mm,
because you want to make a perturbation, but you also want to make the executor as the association with all remaining variable as the same as the original. So this is the constraints. So you can say, so you just solve this um, you know, op optimization problem, then you can get the we call the knockoff operation. So that is the scheme. So with this scheme, you can say do the causal inference.
mm-hmm. Okay.
Okay. So we call the knockoff backdoor criterion. Okay. Infer the interval distribution from observed data. Okay, I think I can pass all the computational
Mm-hmm.
testing. We just
Mm-hmm.
comparing to the many different methodology. So I think I passed those one.
Mm-hmm.
So come to the conclusion. I think we
Mm.
don't have much time. The conclusion is that source is an interventional direct causal inference because we condition all remaining variables. Uh, so the second and feature is the no requirement on the network structure pre-knowledge. So I think that is better than the the backdoor criterion. Another feature is they can do for loop, traditional backdoor criteria, they require, you know, um, network without loop. So, so it also can be used for time series and non-time series.
Mm-hmm.
So, so I think I
Mm-hmm.
finished this one. Oh, another one is I just want to compare to the original backdoor criteria. As I mentioned, backdoor criteria you can see is they just in summarize, I mean, integrate the C, so you can get the new say a new distribution, intervened distribution. Here, because we make another executor, we have to do the twice for the integration, so we can eventually. We sacrifice the computational, and then we can get we remove the constraints for the
Mm.
network. Yeah. So you can see traditionally they require pre-knowledge, they fix the link, backdoor. But then now we assume everyone is a backdoor. That's why we need additional
Okay.
integral. So
So,
just like this one,
so
we build this one, we assume everyone is the backdoor
yeah.
because
So,
we build it. Yeah. So
yes.
it's a
Uh,
quite they have
yeah,
some
yeah, yeah.
relation, but the
So,
scheme is completely different.
so
Okay.
Luonan, so before this slide, you introduced the backdoor thing first time. Uh, before
A
this slide. Yeah.
backdoor criterion is Judy Pearl's theory.
Yeah.
It's a
So
Dubin.
actually, we are
Yeah.
not familiar with that. Can you explain one more time the backdoor, the basic
Oh,
one first?
backdoor. If
Uh,
their network, if you change X, you can see how Y change, right?
uh,
That is a causal inference. If
yes.
you change X,
X.
so you can do X means we fix X to certain value. If you use drag, originally X is zero
Uh,
without drag. With the X is equal one means we use the drag, right? So we can see how disease is affected or not, right?
uh,
So backdoor criterion, but you can use traditionally we just use the conditional distribution, right? We just without do we just the x and conditional x we can infer distribution of y, right? But the problem is this is an association study, not a interventional causal inference. Uh, the reason you cannot change x to any arbitrary value. If
mm,
you change x, you have to cut all the link between the x with c. Otherwise, you cannot change x. So that's why
uh-huh.
even you use the
Yeah. Uh,
conditional
uh,
distribution, you
so,
cannot get the causal inference, right?
sorry. Let me ask you the simple thing first. Can you explain what is the meaning of do here?
Do means change x to certain value.
Okay, so kind of
Mm, mm.
so when
And
then
even this value is not observed.
okay.
The x you have to do means that you change x, you fix x to certain value. Uh,
Mhm.
so it's not the same as the conditional distribution. Conditional distribution is also x at a certain value. We can see y
Then
distribution, right?
why I'm confused is then left side is do x is x can be either 0 or 1 or 2, right?
Right, right, right,
Yeah, but
right.
the right side, your concluded equation is x looks like fixed there.
X. Yeah,
Yeah.
yeah. We fix to x to certain value. We can see their distribution.
Oh, yeah, that's why I'm
Mm,
confused. The left side is depending on the status of the x, but the right
mm.
side final equation,
Oh, but this one.
yeah,
So they can
there's no
be,
do anymore.
we
That's
can,
why.
we use this one. In this one, we can use the observed data to,
Uh-huh.
this one,
Uh-huh.
this is a, this distribution, you have to calculate it from observed data, not intervened data. We
Uh-huh.
don't have intervened data. So,
Hmm.
this the, I mean, this equation, they consider is a revolutionary equation.
Yeah,
So they can use the observed data
yeah,
to get the intervened distribution. Yeah.
because
Uh,
anyway, in this
mm,
equation,
mm,
right side equation
mm,
is
mm.
fixed value because
Yes,
x is
yes, you can.
observed and right side is fixed. So then why
Mm,
I'm confused is left side,
mm.
regardless of the what is it, do x could be 0 or 1 or 2. Their
So you
probability
can fix
is
to
the
any
same.
value. You
Oh,
can
yeah,
do
yeah.
fix to any value.
So,
Yeah.
so that do will be fixed to either x to the 0 or 1 or 2.
Right, right, right.
Yeah, yeah.
But this one x is the same. So it's
Ah,
then we can remove the do. If you
mm,
use the do, use the do, you cannot do simulation
okay.
because you do is a is a then we remove the do, then we can use the observed data to get
Mm-hmm.
this one. So you remove the do is very complicated. This is
Uh-huh.
you need many
Mm,
mathematical manipulation.
okay, okay.
Eventually remove the do, then make them in this way,
Mm-hmm.
so we can use the observed data.
Mm,
This here without do, we just use the observed data, but
okay.
is equivalent to. So it's so I didn't show the theoretical
Mm-hmm.
inference. I mean manipulation. You implementation. You can see the original book. Judy Pearl. So they get the fuse
Mm,
word just for this theory.
okay.
Mm, mm. Okay. So that is sorry. I thought that you are familiar with the
Oh, no, 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 no.
uh,
Sorry, sorry. Yeah. Okay.
okay.
Then maybe we can get a couple of questions from audiences if you
Oh,
have.
we have not yet finished.
Oh, not yet. Oh, I
We have
see.
another
I thought
one.
that you've.
Yeah, we have
Okay.
another one. But you can, if you think it's okay, I finish, stop
Oh,
here.
oh, I see.
Actually,
Oh,
I want to show another recent work, but
oh,
I
okay, gotcha.
think maybe we don't have time. We
Okay.
just stop here. Yeah.
Okay.
Mm.
So maybe, yeah. Is there any question from audiences? Oh, because I thought that you showed the conclusion so that you're done.
Oh, yeah, this is the first one.
Oh, okay.
We have another one.
Okay.
This is mainly for the sample-based data. Another one is time course-based
Uh-huh.
data.
Okay.
So method is different. But
Okay.
yeah, I think maybe next time I will
Yeah, yeah, yeah.
show you
Okay.
that. Mm, mm.
Can I ask a question?